The overall goal of this procedure is to create 3D tissue constructs from aggregated cells and cell-derived matrix that can be used for functional analysis. This is accomplished by first milling a polycarbonate mold to create round-bottomed annular wells. Next, polydimethylsiloxane, or PDMS, is cast onto the polycarbonate mold to create a template. Then, the PDMS template is used to form agarose wells. The final step is to seed cells into agarose wells to allow them to aggregate and form cohesive tissue rings. Ultimately, this method can be used to achieve cellular self-assembly and tissue engineering using different cell types, and enables analysis of the contribution of cells and cell-derived extracellular matrix to tissue structure and function.
The main advantage of this technique over other tissue engineering approaches is that we can generate tissue constructs entirely from cells and the extracellular matrix that they produce more rapidly than previously reported and in a format which is conducive to mechanical analysis.
Begin by milling a half-inch thick piece of polycarbonate to create 15 round-bottomed annular wells with a center post diameter of 2 mm. Clean the polycarbonate mold to remove any plastic debris generated from the milling process, and let the mold dry. Next, mix PDMS at a 10:1 ratio of base to curing agent. Degas the solution to remove all air bubbles, and then pour it into the polycarbonate mold. Then, degas the mixture on the mold again to remove any remaining air bubbles, and cure the PDMS in an oven at 60 degrees Celsius for 4 hours. While the PDMS is curing, dissolve 2% agarose in DMEM. Once cured,